Hello dear students, in this video we'll continue chapter 7 section 2. We will talk about test cross, incomplete dominance and codominance. What is test cross? It's a cross that's used to test what or to determine what. It's used to determine the genotype of what, of unknown what. Is it of unknown dominant or recessive? Of unknown dominant trait. So we have a phenotype of a dominant trait, like for example, let's say we have purple flower. This is a phenotype of what? Of dominant. And we don't know if this purple flower is it P capital P capital or P capital P small. So what we can do in order to know which one is it, if it's P capital or P capital P small, it's we will do the test cross for it. So this test cross, is it to know the genotype or phenotype? It's to know the genotype of what? Of dominant or recessive, of the dominant trait and non-dominant trait. And how we will know it? By what? By crossing it with what? By crossing it with recessive trait. So we have unknown genotype, we will cross it with the homozygous recessive or the recessive trait. And we will focus on the offspring after that. For example, usually we can do the test cross for any of the Mendelian genetic, of the complete dominance of Mendelian genetic. What we mean by that, for example, if we go to the flower of the pea plant, for example, we talk now about the flower of pea plant. We said we have purple and we have white. For which one we can perform the test cross? For the purple, because we said for the dominant phenotype, it's not for the recessive, because the recessive we already know it. For example, we can do the test cross for this round seed, because this round seed we don't know if it's R capital or R capital R small. At that time we will do the test cross for it in order to know if it's homozygous or heterozygous. The same thing we can perform for the other characteristic of the 7 character of the pea plant. We can do the test cross for all of these what of the dominant trait, because it's for the dominant, it's not for the recessive. So this tall plant, because the tall stem pea plant, we don't know if it's T capital or T capital T small. So to check the purity of this tall pea plant, which means if it's homozygous or heterozygous, we can perform the test cross for it. The same thing for this plant. This plant have the flowers, its axial position. So in the position of their flower, the axial one might be A capital A small or I capital A capital, might be hetero or homo. So how we can know if it's hetero or homo or check the purity of this dominant trait? By test cross. The same thing for purple flower, because it might be P capital or P capital P small. Also for the green pod, which is also a dominant trait, might be G capital or G capital G small. For the inflated pod, it's also a dominant, so it might be I capital or I capital I small. The same thing for the yellow seed. Yellow seed, it's dominant and it might be Y capital or Y capital Y small. For the round seed, the same thing. It might be R capital or R capital R small. So we can perform the test cross for all of these dominant traits of the Mendelian of the pea plant. But for the recessive ones, we can perform the test cross because already we know their genotype. For example, we know already that wrinkle seed, it's R small. And the green seed, because it's recessive, so it will be Y small. And for the constricted, it's also recessive, I small. For the yellow pod, it's the recessive of the pod color, so it will be G small. What about for the white flower? For the white flower, it's P small. And what about for terminal? You know the terminal. Please pay attention to this figure. You should memorize each one and if it's dominant or recessive. For example, if you see the flower in the top, this is for top for the terminal. So it's a recessive trait of the flower position, because flower position, if it's in the side here, it's axial, and if it's in the top or in the tip of the flower, it will be terminal, so it will be a small. What about for short? It's T small. So you can see that for the recessive trait, you only have one genotype and you know it. But the one that might be unknown, and you can choose, you can determine if it's, for example, T capital T capital or T capital T small, at that time you can do what for it? You can do a test cross for it in order to know if this tall, the purity of this tall, is it homozygous or heterozygous. How? By crossing it with recessive trait of it. Okay, so test cross to determine what? It's for genotype or phenotype to know the genotype of dominant or recessive of the dominant one. How? By crossing it with recessive. This is the rule for it. Now let's take an example for it in order to know how to solve the kinds of question or the solve questions about test cross. In a cross between a black guinea pig, we have a black guinea pig, but still we don't know if this black is B capital B capital or B capital B small. Okay, and the cross happened between black guinea pig with a brown guinea pig. You should memorize that the brown guinea pig it's B small, while the black one it might be B capital or B capital B small. Now he said that half of the offspring was brown, so 50% of the offspring brown. And you know the brown is recessive, it's B small. So at that time, and as we said in the previous video, when you see that half of the offspring are recessive, so one of the parent recessive and the other one will be homo or hetero, it will be hetero. Because when you cross this one with this one, you can delete the repeated one. So we will get B capital B small and B small. So we will get half of them are B capital B small and half of them are B small. So whenever you see that the offspring, half of them or 50% of them are recessive, so the parent, which is unknown, is it homo or hetero? It will be what? It will be hetero. Okay. As you can see here, at that time will be hetero if you have a ratio of 50%. Like here in the cross, as we can see here, if we did it as a Punnett square, we have B small. If we cross it with B capital B small, at that time 50%, as the question said, 50% will be B small, or will be in brown color. The other example that we have in a cross between a black guinea pig, still as we said before, the black we don't know if it's B capital or B capital B small because it's a dominant. And we cross it with a brown guinea pig. The brown is B small. All of the offspring were black, so 100% they are black. Okay, and there is no other phenotype, there is no brown in it. So at that time, if you get 100% or 1 to 0, the dominant trait appear in the offspring. So at that time, it means our parent is what? The parent will be B capital. It's not B capital B small. Why? If you cross this B capital with B small, so at that time, all of the offspring will be what? All of them will be B capital B small. Here we have B small with B capital, so all of the offspring will be B capital B small. It means all of them will be black. So whenever you get 50% in the offsprings are 50% in the offspring are recessive, for example T small, it means here we have this parent will be what? Will be hetero, because already we cross it with recessive. But when we get 100% are dominant, at that time it means, or hetero, at that time it means we have homo with homo. It means with the recessive one we will cross what? We will cross the dominant one. It will be homodominant. Now we have some notes here. We can do the test cross for all the dominant trait because we said that test cross is used to know the genotype of what? Of the dominant trait. Because for example, we don't know if this dominant trait is Y capital or Y capital Y small. So at that time we can perform the dominant test cross for it in order to know its genotype. And for the recessive trait and for incomplete dominance that we will talk about it later, there is no need to perform the test cross for them because already we know their genotype, so there is no need for the test cross. Okay, another note we have, test cross can perform to check what? To check the purity of the dominant. We don't know if this dominant is homo, which means pure, or heterozygous, so we do the test cross in order to check their purity. From all the flower colors that we have, only purple pea plant we can do or perform the test cross for it, because it's the only dominant with unknown genotype, which may be either P capital P small or P capital P capital. What we mean by that? Only for purple flower. For example, if you look to the flower colors that we have, we will talk later also about these flower color. We have white flower color, we have red flower color, we have pink and purple. The white flower color, we will talk about it. It's incomplete. This is the pea plant, and it's p small. So it's recessive, and there is no need for the test cross. And for the red and the pink flower, we already know their genotypes, and we will talk about it later. There is no need to perform test cross for them. But what about the purple flower of pea plant? The purple flower, it might be P capital or P capital P small. So at that time we need to perform what? To perform the test cross in order to know if this purple it's P capital or P capital P small. Now, question one about test cross, if which of the following is a result of test cross? Usually when we talk about test cross, you should focus on something, which is one of the sides should be recessive, because we said that we will cross this dominant with a recessive trait. So which one here consists of contain recessive trait? Only this one, because here we cross it with what? With t small. Only this one can be a result of the test cross. Question two, the genotype of a dominant which is unknown. As we said, we have dominant unknown, and we want to know the genotype of it. It can be homozygous if the ratio of the resulting offspring from the test cross is what? He said that we have test cross here, and here we have genotype of unknown. We know that already we will cross it with recessive, for example, R small. At which time this one will be homozygous, it means R capital, when the offspring 50% or 100% will be dominant. Sure, it's when 100% will be dominant. At that time, we can say that this parent is homozygous. Question 3, in test cross, the crossing occur between individuals that have dominant trait with another one that have what? We cross the dominant in the test cross with a recessive trait. So we will choose recessive trait. The unknown genotype of an individual with a dominant phenotype, we have phenotype dominant, and we don't know the genotype of it, if it's homozygous or heterozygous. It can be determined by what? It can be determined only by what? By using the test cross. So we will cross it with recessive in order to know if it's homo or hetero. Question 5, if the unknown parents, so we have two unknown parents, they are crossed, and the offsprings contain 25% recessive. So both parents must be what? We said whenever you see 25% is recessive, it means the parent should be hetero. Both of them should be hetero, or we can say hybrid. So both are hetero and both are hybrid, so we'll choose E for it. Now we will talk about the three kinds of dominance. We talked about the first one. We said that it complete dominance. It's Mendelian genetic. What we talked about it before about pea plant, about guinea pig, and the rabbit coat color. All of these were Mendelian genetic and Mendelian traits. We said we have complete dominance. They named as complete dominance. And why they named as complete dominance? Because if we have the recessive trait and it's come with the dominant trait, the recessive allele, and it's come with the dominant allele, at that time the dominant will completely cover or mask or hide the effect of the recessive allele. That's why it's named as what? It's named as complete dominant, because it dominates on it, and at that time it appears by the dominant. We say it's purple. Okay. In complete dominance, we said we have only two traits. For example, you can look here. If we cross this blue with this red, only one of the parents' phenotype will appear in F1 generation. It means it appear blue. For example, let's say this is the dominant and this is the recessive. So the dominant one will appear in F1 generation. This is incomplete dominance. Why? Because the dominant will cover or mask the effect of the recessive. That's why it appears only by the dominant trait in F1 generation. And this is incomplete dominance. As you can see, we have only two colors. So we say that we have only two traits. To control contrasting trait. For example, in pea plant, we said we have purple and white flower. In incomplete dominance, neither allele is completely dominant over the other. And here, the result here or the offspring will be intermediate or in between. So incomplete, it means intermediate or in between the two contrasting traits of the parents. So as you can see here, another trait will be produced. So this trait, this is the third trait, because here we have a color and this is the second color and this is the third. So we have three traits for each character, and this one, the third one, it will be should be heterozygous, and these two parents should be homozygous in order to get this heterozygous, and this one will be intermediate or in between the contrasting traits of the parent. For example, if you mix the blue with the red, so at that time you'll get purple. So this is when you blend or mix both these two parents, and the result will not resemble any of the parents. It's not look like any of the parents. Instead, we mix or blend both of them, and we get another trait which is intermediate or in between the two parents. This named as incomplete dominance. Okay, what about this is in shortly, we will go to talk each one in detail after that. What about in co-dominance? In co, it's cooperate. So both of the parents cooperate, both of them express themselves in a heterozygous condition. We can see, for example, if we cross this blue with this red, so at that time both of them want to express and want to show themselves. So that's why both colors appear in it, and they don't blend together, they don't mix together. Instead, both of them express themselves, and both of them shows themselves, and that's why it's named as co-dominance, and they cooperate in order to express themselves in the offspring. Now, returning back to talk about each one in detail, in complete dominance is named as Mendelian trait. Incomplete and the codominance, they are non-Mendelian. So we will use different letters from those of Mendelian genetic or Mendelian traits. Incomplete dominance or the Mendelian trait, it's a relationship, we can say, in which in the heterozygous condition, for example, heterozygous, like for example, P capital P small, which is for purple flower, one allele it's completely dominant over the other. That's why it's named as complete dominance, because here we have an allele like this one, the capital letter or the dominant allele will dominate or cover or mask the effect of the recessive one. That's why it's named as complete dominance. In complete dominance, the heterozygous plants, for example, P capital P small, and the homozygous dominant plant, like P capital, this is homozygous, are indistinguishable. Very important. When we talk about complete dominance or Mendelian trait, we can distinguish between P capital P small and P capital, because both of them are purple and both of them show the dominant trait, and you can distinguish between them. So at that time we can perform, for example, the test cross in order to know if the dominant is P capital or P capital T small, P small. This is another phenotype of them. We can distinguish between them, not in their genotype, because in their genotype we already know their genotype. If he give you the genotype, you can know the phenotype from it. But from pheno, we can't know it's a geno, okay? And we can distinguish between them in the phenotype. For example, as we said before, we have P capital and P capital P small, both of them for the flower color, and both of them have the purple color. Now, how you can identify or how you can know that the question talk about complete dominance or Mendelian traits? First, if the question asks about pea plant, and you know pea plant is Mendelian genetic and it's complete dominant, all the 7 characters of Mendel that Mendel studied, and also the coat color in rabbit, as we said before, we have black and brown, and the guinea pig, also we have black and brown. So when any question asks about any of the character of the pea plant, of the coat color in rabbit or the guinea pig, we will perform the rules of Mendel. And the rules of Mendel, as you know, that for example, if I talk about purple, purple is dominant, and for dominant we have 2 genotypes, either P capital or P capital P small. And for the recessive one, which is white flower, I will write it p small p small. So it's the same first letter of the dominant and I put it in small letter, small letter. These are the rules of Mendel's. Okay. If we see a dominant allele in the question, he said that the dominant allele hide the effect of the recessive allele. As we know, the dominant hide the effect of the recessive allele in the heterozygous condition. So this is also for what? For Mendelian genetic. We will apply the rules of Mendel. Okay, and also if we see only two traits for one character, at that time you will know it's Mendelian genetic again. Why? Because we talked about P. plant, for example. We said that Mendel said that he studied each character with two contrasting traits. For example, round and wrinkle, green with yellow, inflated with constricted. So if we see only two traits, so this is also for each character. So this is also will be complete dominance and Mendelian genetic. The rules for complete dominance, as you know all, that for example, if I cross black with white and this one will be dominant and this one will be recessive, so the black will be B capital, for example, and the white will be B small. So we should use the same letters of what? Of the dominant one, okay, of the first letter of the dominant. And at that time from this cross, 100% will be what? Will be black, because we don't have another trait here. It's only we have two traits for each character, black and white, and as a result one of them will appear, either the black or the white. Which one is dominant, it will appear in F1. What about in incomplete? Incomplete dominance, it means intermediate or in between. So another character will appear which is in between or intermediate between the two traits of the parents. For example, the definition for the incomplete dominance, which is a type of non-Mendelian trait, we will use different letter and different rule of from Mendelian or from Mendel's rule. It's a relationship that occurs when the phenotype of the heterozygous, the phenotype here of the heterozygous is intermediate or in between the phenotype of what? Of the two contrasting traits of the parents, the dominant homozygous and the recessive one. Okay, here neither allele, we can't say that we have allele is dominant over the other allele. Let's take an examples of the incomplete dominance. First, we said that the rules for incomplete dominance is differ from Mendelian genetic and from complete dominance. Here we will use different letter and we will use only capital letters. For example, if you cross black with white, here we will say that black is B capital, and the white we will use different letter. Depend on this one, it's WW. It's not like Mendelian genetic. In Mendelian, we said it's B small. So if the question is incomplete, how did you know it's incomplete? For example, if he said that when you cross black and white, another trait appear which is gray. When we mix black and white, what will happen is that the result of this mixing or blending of these two colors, it will be gray. This gray, it's intermediate or in between the two traits of the parents, which are homozygous dominant and the homozygous recessive. So you can say that this is will be intermediate between these two color. That's why it's incomplete dominance. And because it's incomplete dominance, so the black will be B capital, and the white will be WW. And we cross both of them, we will get what? We will get BW, for example. Now, how you can identify that this question asked about incomplete dominance? First, if you see in between, intermediate phenotype, if you see a phenotype which is intermediate or in between the dominant and the recessive one, at that time we say it's incomplete dominance. For example, you should memorize these examples, please, for the incomplete dominance. We have the pink color flower and the 4 o'clock flower or snapdragon flower. So we have a flower which is named as snapdragon or 4 o'clock flower. This flower, we have three colors for it. We have white, we have red, and we have pink. Which one is incomplete or intermediate? The pink is intermediate or in between the white and red. So at that time we say that the white color will be WW. What about the red? The red will be RR. And what about the pink? The pink is intermediate or in between both of them. So in between both of them, it will be RW. Also, we have the gray color. The gray is between white and black. So whenever you see a color or anything which is intermediate or in between or is a result from blending or mixing two other colors, so at that time we say it's incomplete dominance. The gray color, for example, also the same thing. The black will be B capital, B capital. The white will be WW. And if you cross both of them, we will get gray color. And the gray is intermediate or in between both of them, so it will be what? It will be incomplete dominance. Incomplete dominance, it's this trait, for example, which is it will be in heterozygous condition, and the phenotype of it will be intermediate or in between the phenotype of the parent. Also, we have another example which is very important, the American short tail of the cat, which is between long tail and no tail. We have, this is for long tail, LL, and the no tail will be NN. So if you cross long tail with no tail, we will get what? We will get LN or NL. This will be for the short tail in the cat. The short tail is intermediate or in between the long and no tail, so we say it's an incomplete dominance. Also, we have the wavy hair. This is for chapter 9. It's not included this year, but it's also easy. We have the wavy hair. It's between straight and curly. So if you cross straight with curly, straight is SS and the curly is CC, so SC or CS. This will be wavy. This is an intermediate between the straight and the curly. If we see also in the question, two allele blend effect or mix together, each in each other, they blend with each other in a heterozygous condition, so this will mean also incomplete dominance. So we need the intermediate should be heterozygous and blend the effect of both the parents. Always the phenotype and genotype ratio are the same. Very important note. Here we will focus on the genotypic ratio and phenotypic ratio. In all incomplete dominance, the phenotypic ratio and genotypic ratio are equal to each other. So if you know the phenotype, it's directly equal to the genotype. Okay, and the vice versa. Or also if in the question you see three traits for each character, because you know in Mendelian P-plant or in the traits, the Mendelian traits, we have only two traits. But here we have three traits for each character, such as, for example, if I talk about the flower color of Snapdragon, I have pink, red, and white. And also when we have two or more alleles influence the phenotype. If two or more alleles influence the phenotype and result in a phenotype which is intermediate between the dominant and recessive, this one will also indicate that we have incomplete dominance. Now we talk about the incomplete dominance, and incomplete, we said that we have in between or intermediate phenotype will appear, and it will be heterozygous between two contrasting homozygous parents. For example, if we cross we said an example for it, it's Snapdragon flower. This flower, we have three colors for it. We have the red color. Let's do it as red color. We have the red one, and the red, we said we will name it as R capital, R capital. What about if we cross it with the white flower color? And the white flower color, let's name it as WW. We have white flower, it's WW. So whenever we cross red with white, what will appear is that all the offspring here, as we said before, we can delete the repeated one, and all of the offspring will be what will be RW. RW or WR, it will be with pink color, okay, because for the Snapdragon flower we have three traits for it, or we have three colors for it. We have the red flower Snapdragon, we have the white flower Snapdragon, and we have the pink. Now, which one is intermediate, or it results from blending or mixing the two parents? It's the pink color, which is RW. As you can see here, the pink, it's intermediate or in between the white and the red one. And it's in a heterozygous condition. What about the parents here? They are homozygous. Both of them are homozygous. And here in incomplete dominance, we can see that we have three different colors. So it means we have for each character, we can have three different traits with three different phenotype and three different genotypes. If we do like what Mendel did in F1 and F2 generation, for example, in Mendel, he crossed homodominant with homorecessive. So he crossed what? For example, he crossed the purple flower homodominant with white flower, which is a homorecessive. And what he get in F1, he get all of the F1 are P capital P small, which mean all of them are dominant. And if we do it the same here in the incomplete dominance, here we cross homo with homo, like for example, red flower, R capital, with the white flower, WW. And what we get in F1 generation, we can say that we get all of the offspring will be RW. So the same thing here, all of them are hetero, and 100% they are what? They are hetero, and they are in pink color. Okay. Now, what about in F2 generation? If we cross in F2, what Mendel did, he crossed the P capital P small by itself, which is self-pollination. He did self-pollination for the F1 generation. And what he get from here, it's 1 to 1. This is the genotypic ratio. And the phenotypic ratio is 3 to 1, 3 purple to 1 white. What about here in the incomplete dominance? In the incomplete dominance, also if we cross this RW with another RW, which means we will do self-pollination for the F1 generation. If we do self-pollination, as you can see here, if we do self-pollination for this F1 generation, which is RW, so we cross RW with RW, we also will get 1 to 2 to 1. And we will get one of the offspring will be red, two of them will be RW, which is pink, and one of them will be WW, which is white. So for the phenotype and the genotype ratio, it will be 1 to 2 to 1. We will get the same ratio because we said before that in incomplete dominance, the phenotype is equal to genotypic ratio. Pay attention to that note, okay? We can't see the ratio of 3 to 1 here in incomplete dominance. We never see this ratio. We only, for example, we can see another ratios, but 3 to 1, we never see it in incomplete dominance. And in incomplete dominance, we can see 3 traits. For example, let's talk now about another example, which is the tail in the cat, the length of the tail. We said also for the length of the tail of the cat, we have 3 traits. What do you mean by that? We have long tail, and if you cross it with no tail, which is, you can say, the opposite of it, at that time we will get what? Intermediate, or in between the no tail and the long tail, we will get short tail. So now which one of these 3 traits is incomplete dominance or intermediate? It's this one, the short tail. The short tail is intermediate or in between the long and the no tail. If you blend or mix both of them, we can get the short tail. So you should memorize each one with its genotype. For example, the long tail, usually it's LL. For no tail, it's NN. And if we cross LL with NN, we will get the short tail between them. So we will get LN. You can do it also by the Punnett square. You have LL and NN. At that time, what we will get, all of them will be what? Will be LN, which is the short tail in the cat. And this one, it's intermediate, or we can say in heterozygous condition, and intermediate or in between the two parents of contrasting traits. So in the snapdragon flower or 4 o'clock flower, we have these 4 possibilities. If we cross red, this is red, so R capital with white, so it's WW. At that time, when we mix red with white, we will get only pink flower color. So all of the offspring, 100% will be what? Will be pink, which is, you should memorize it, it's RW or WR. If you cross pink with pink, it means RW with RW. At that time, you will get all the 3 colors. We will get one of the offspring will be red, 2 of the offspring will be pink, and one of them will be white. So you can get all the 3 colors. If we cross pink with pink, at that time, you can get red, white, and pink in a ratio of 1 to 2 to 1. This is for both phenotype and genotype. What about if you cross white WW with pink, which is RW hetero? Whenever you cross homo with hetero, we said half of the offspring will be hetero like this parent and half of them will be like this parent. So we'll get 50-50 ratio as 50%, 50%, 50% white and 50% pink. So we can say 1-to-1 ratio. And if you cross white with red, the same thing, because this one it's WW, it's homo, and the white with red. And if you cross white with pink, sorry for this one, it's red and pink. The red one it's R capital, which is homo, and the pink is RW, it's hetero. Whenever you cross homo with hetero, so half of the offspring will be from this hetero one, which is pink also, and half of them will be what will be R capital. You can do the cross and you can see the same thing. We have RR and RW. So the RR will be red and the RW is pink. So we have half of it will be pink and half of it will be red. So whenever you cross red with white, this is like F1 generation of Mendel, because homo and the result it's all hetero and 100% it's hetero. It's the heterozygous. What about if you cross pink with pink? This is like F2 generation of Mendel when he did self-pollination. Between the hetero and hetero, we get 1-to-2-to-1. But here we have three different rates with three different phenotype and three different genotype. And if you cross homo with hetero, we can get half of them hetero and half of them homo. If you cross white with pink, half of the offspring will be white and half of the offspring will be pink. If you cross red with pink, half of them will be red and half of them will be pink. This is the incomplete dominance, and incomplete dominance it means the intermediate or the in between the color that is result which have heterozygous condition, it's in between the two homozygous parents. And the one which is incomplete, it's only the pink color here. If the question asks in which of the possibilities you can get pink flower, if you cross red with white, yes, we can get pink flower. If we cross pink with pink, yes, we can get also pink flower. If we cross white with pink, we can get pink. If we cross red with pink, also we can get pink. But in which one we can get 100% pink? If we cross red with white. In which one we can get 50% of the offspring pink? Here we have 50% pink if we cross pink with pink. And also here we have 50% pink if we cross white with pink. And here we have also 50% pink if we cross red with pink. So here we have some examples of the incomplete dominance. As we said before, if we cross pink with pink, here we have RW, which is for pink flower, Snapdragon, and RW. So as you can see, one of them will be red. This is red color, RR, and two of them will be pink, RW, and one of them will be white. So we can get this ratio 1-to-2-to-1. This ratio for both genotype and phenotype, they are the same in incomplete dominance, because as you can see here, one of them will be red, one of them will be white, and two of them will be purple. And the same thing for their genotype. We have RR for the red, we have 2 RW for the pink, and 1 WW. Okay. As a result, we can see that we have three different phenotype and three different genotype for each one that is produced. We can talk about some differences between incomplete and codominance. In incomplete, we said that we have a phenotype that appear, it's in heterozygous condition, and it's intermediate, incomplete, intermediate, or in between the two phenotype of the parent. Incomplete dominance, we said it's a relationship when the heterozygous condition, the dominant allele completely dominate or cover the mask or effect of the recessive allele. Here we have three traits. For example, we said in flower Snapdragon, we have red, pink, and white. But for the complete dominance, we have only two traits. For example, for seed color, we have, for the flower color, we have the purple and white. For seed color, we have green and yellow. Please correct this to the flower color. Okay. The incomplete dominance, it results in phenotype which is intermediate or in between the dominant and the recessive. So we have neither allele is completely dominant over the other. While in the complete dominance, the dominant it's cover or mask the effect of the recessive. This we can only see it this statement in complete dominance when we say dominant, so it completely dominate or cover the mask of the effect of the recessive. F1 generation, it does not resemble any of the parents because as we see, for example, when you cross red with white, so the result will be pink, and this pink it's not resembling any of the parents. It's not look like, for example, the first parent or the other parent. Okay, it's completely different from both of them because we blend or mix both them and we get another trait which is completely different from them. While F1 generation, here in Mendelian experiment, it resembles one of the parent. As you can see, if this is the dominant, which is the purple pea plant, and this is the recessive white flower pea plant, so F1 generation will resemble, it's look like one of the parents' phenotype. Pay attention, not the genotype. It's look like the parents' phenotype. And the F2 generation, as we said before, we have 3-to-1 ratio. Three of them will appear with the dominant trait, for example purple, and one of them will appear by white color, which is the recessive one. Okay, now the phenotype of the heterozygous, as we said, it's intermediate between the phenotype of the contrasting trait, and we can distinguish here easily between them. So there is no need for test cross because easily you know that the red it's R capital, the white it's WW, and the pink it's between them and it's RW. So here there is no need for test cross, and we can distinguish between them. But in which one we can distinguish between them and it's indistinguishable phenotype? This is in complete dominance, in Mendelian genetic, because we said that the heterozygous and the homozygous dominant, they are indistinguishable in their phenotype. What do you mean by that? It means P capital P small, this is the heterozygous, and the homozygous dominant, which is for example P capital, you can distinguish between them in their phenotype. Both of them will result in purple flower, while in incomplete, yes, you can distinguish between all of them. For example, here we can't know if it's this P capital or P capital P small. Okay, but if we talk about F1 generation, yes, at that time this one will be homo pure, and this one will be P small, while the F1 generation, all of it will be heterozygous P capital P small. As you can see here, please pay attention to these flowers. This type of flower is for what? This type of flower, it's pea plant. Okay, this one it's pea plant, and this pea plant it's white flower, so it's P small. What about this one? This one that is like that, they are for snapdragon, and the snapdragon flower, the red one will be R capital, the pink flower will be RW. What about the white snapdragon? White snapdragon look like that, it's WW. Pay attention to the white pea plant and the white snapdragon, like their shapes. This one will be P small if it's for the Mendelian genetic or pea plant, while the white flower snapdragon it's WW. Please pay attention to them. And what about the purple flower? Purple flower may be either P capital or P capital P small. So which one of these four need test cross? If it's unknown, we have unknown dominant genotype. We already know these the genotype of all these flower, but only the purple flower it might be P capital homozygous or P capital P small, which is heterozygous. So we can perform the test cross only for this one, which is for the purple flower pea plant. If we compare the F1 and F2 generation of Mendelian, what Mendel did, he get the P generation, as we said before, by true breeding. He crossed two contrasting traits, one of them purple flower with white flower. What he get in F1, all of the offspring will be purple, will be dominant, and will be hybrid. And when he do self-pollination for this purple flower, in F2 generation, he get a ratio of 3-to-1. This is for Mendel. What about for the incomplete dominance? For the incomplete dominance, if we cross the pink flower with white flower, if we cross the red with white, we can get all of them will be pink. And if we cross pink with pink, this is like F2 generation. If we cross the pink flower, RW with RW, both of them hetero, so we can get one of them is red, two of them are pink, and one is white. So 1 to 2 to 1 ratio. Here you have some questions about the incomplete dominance. You can solve them. And if you have any question, let me know, please. Now, let's start with the last one, which is codominance. In codominance, we say that both of them express themselves in a both of them, express themselves. Codominance mean cooperate. They cooperate, and both the parents, they want to express themselves. They want to show themselves in their offspring. So the offspring will appear by the two traits of the parents. They both express themselves in it, and that's why we can see, for example, if you see that black white coated, black white spotted, whenever you see the dotted, spotted, or for example for the cow, for the cat, all of these spotted and dotted will be what will be codominance. Codominance, it's another non-Mendelian trait, which means we will apply also non-Mendelian rules in it, not those rules of Mendel in it. And it's also a relationship that occur when both alleles for a gene are expressed. They both of them express in a heterozygous offspring. So again, the offspring will be heterozygous, and but here it's not blend. We don't blend or mix both the parents together. It's not intermediate or in between the two parents, but here both of them express themselves. Okay. For example, when we talk about incomplete, we say if we cross black with white, it will appear gray. But here black with white will appear black white spotted. So very important to know if the question asked about which one, is it incomplete, is it complete, or codominance. When the question said both of them express themselves and they don't blend or they don't mix or it's not intermediate, this one will be codominance. It's not incomplete. Incomplete, you have intermediate or in between. For codominance, they cooperate and both of them express itself in a heterozygous condition. Here, neither allele is dominant or neither allele is recessive. We don't have one is dominant and one is recessive because both of them express themselves and they don't blend together. Very important. The rules for codominance, it's like the incomplete dominance. For example, we will use different letter and capital. For black, we say it's B capital, B capital. For white, again, it's WW. But what appear, it's BW or WB. It's a heterozygous. It's not gray between the black and white. It's not incomplete. This one will be black white spotted. Both of them express themselves in a heterozygous condition. So how can we identify the codominance in the question? If we see, as we said before, both alleles show their effect or express themselves in a heterozygous condition. If the question says both allele, they don't blend together and they both affect in heterozygous condition. An example for it, for this year only MN blood type, we have AB blood type and MN blood type. The MN blood type is an example of a codominance. In the codominance, the MN blood groups, we have another type of blood group. As we said before, we have ABO blood group. Now we have also another type of blood group which is named as MN blood group. In this MN blood group, also we have the RBC, and on the surface of RBC, some people have a molecule which is named as M molecule, and their blood group is M. And other people have on the surface of RBC another molecule which is named as N, so their blood group will be N. But if the person have both of them together, have N and M molecule on its RBC, on the surface of RBC, so at that time their blood group will be Mn. So we have three different phenotypes because we have three different blood type for it. We have three different phenotypes: blood group N, blood group M, and blood group Mn. And also we have three different genotypes for it as well. But for the alleles that determine it, it's two alleles only. We will look at it now. Let's take an example first. A black coat color cat. We have black coat color cat. It's crossed with white coat color cat. And among the offspring, a black white spotted. So when you see black white spotted, directly you should know that both parents, black and white, they cooperate and both of them express themselves. So it's a codominance. So at that time, we say the black. We don't have B capital, or B capital, B small. Directly, you should say it's only B capital, because we in codominance, we said that we use different letter and only capital letter. We don't have recessive here. So directly we know that it's B capital, and the white will be WW. And if you cross B capital with WW, you can see that all offspring will be what will be the BW. BW will be black, white spotted, or black, white coated. What about the F1 generation, the phenotypic and genotypic ratio? Here, also the same of incomplete dominance. It means the genotype ratio equal to phenotype ratio. For example, here you can see that all of them are BW. So all of them are 1 to 0. It means 1 to 0 BW. What about the phenotype? All of them will be black, white spotted. So also 1 to 0. This is the phenotypic ratio. So phenotype equal to genotype. This rule will be applied for both incomplete dominance and codominance. For both of them, we have the same rule. But for the complete dominance, not always the phenotype ratio equal to genotype ratio. The MN blood types, they are, we said they are another blood type group. As we said before, beside the ABO blood type, we have MN blood type also. There are three types of the MN blood type. We have M blood group, N blood group, and MN blood group. As we said, if in the surface of red blood cell they have M molecule, so their blood group will be M. If they have N molecule, the blood group will be N. And if they have both of them, M and N, so the blood group of this person will be MN. Which one is codominance? This one. Okay. The phenotype of the blood type M, it's M, and for N, it's N, and for the phenotype of the blood group MN, it's MN. So we have how many phenotype? We have three phenotype. And how many genotype? We have also three genotype. For example, for blood group M, you should represent it by like that, LM, LM. For blood group N, it's represented by LN, LN. What about for blood group MN? It's LM, LN. This one will be codominance. And how we get it? If we cross blood group M with blood group N, you can get, for example, blood group MN, which is a codominant. So now we know that we have three phenotype for the codominance, MN blood type, and three genotype for it. What about it determined by how many allele? Very important. All of the character that we studied, they are determined by two allele. The P plant characters determined by two allele. The incomplete dominance determined by two allele. The codominance determined by two allele. Here the two alleles are what are LM and LN. We don't have another allele. It's either LM or LN, or the person may have both of them together. At that time will be with blood group MN. So very important to know that all the characters that we studied are determined by how many factor or how many allele? By two factor or two allele. The P plants is determined by two factor, either the capital letter or the small. So dominant allele and recessive allele. The incomplete dominance also it's determined by two allele. For example, for the flower color of snapdragon, we have RR, which mean we have this factor, and we have WW, we have this factor. And the person may be like RR, WW, or RW. So it's also determined by two factor or two alleles. And the same thing here for the codominance, also determined by two alleles, either LM and LN. The question about it, if we cross, for example, pairs of codominance, when he say pair of codominance allele, it means we said that we have only one example of the pair of codominance allele for the MN blood group, which is MN, if we cross MN with MN. So at that time, what is the result of this cross? When you cross MN blood group with MN blood group, we say it's pair of codominance allele, and how many different phenotype and how many different genotype will be produced. So if you cross a pair of codominants, it means one person would have which have blood group M with another person which has blood group N. At that time, this is not a pair of codominance allele. This one, for example, if you cross M with N, at that time we will get all the offspring will be with blood group MN. And now if we cross pair of codominants, it means MN with MN, LM, NN. So at that time, what we can get one person with blood group MM and 2% with blood group MN, with 1% that have blood group NN. So we can get ratio of 1 to 2 to 1. So we can get three different genotype, as you can see here. One of them is MM, two of them MN, and one of them NN. So we have three different genotype. And the same thing, 1 to 2 to 1 for the phenotype of them, which we have one of them will have a blood group MM, two of them have blood group MN, and one of them have blood group NN. So if we cross a blood group M with the blood group N, at that time you can get what you can get the blood group MN. This one, it's codominance. Both of these parents, they express themselves in heterozygous condition, and we get this offspring, which is all of the offspring here will be with heterozygous condition, and all of them will be with MN blood group. If we cross a pair of codominants, it means blood group MN with blood group MN. At that time, we can get how many different phenotype and how many different genotype? Three different phenotype and three different genotype. Three different phenotype, the blood groups that we have, blood group M, blood group N, and blood group MN. What about the three different genotype? They are these, the letters LM, LM, this is one, and one is LM, LN, this is the other type, and one is LN, LN, this is the other type.